So my name is Ellen, and I'm going to present the results of my master's thesis, really. It's going to be quite a simple version, I suppose. And it was about detecting 2 seahorse species in Sweden with eDNA, like creating the method of detecting them. And I did this project along with my supervisors Björn and Leon, and I also got a lot of help from Jorge with sampling. And let's start. So first, for an introduction, we've already mentioned this quite a bit, but there's something known as northward range expansion, and that is when the distribution of species change because of climate change as well. The temperature actually pushes them northward due to their temporal maxima, basically. And Sweden is in a very northern country, as we've seen before. As a result, we can kind of expect these more southern species to come into our ranges. And seahorses have already been shown in modeling to have a high potential of coming to Sweden in the future. And it is actually the case that they have already appeared there many times. There have been many observations of both species, Hippocampus hippocampus and Hippocampus guttulatus. Out of those, only 2 can be confirmed to be Hippocampus hippocampus with picture evidence, and about 5 to 8 can be confirmed to be Hippocampus guttulatus. However, I'm sure some of you are thinking, how does a few vagrant species vagrant observations actually relate to the occurrence of species in a country? And that is super hard to answer. And as we heard before, seahorses are quite cryptic in the sense that they are small, they're very camouflaged, and they have this very sedentary behavior. As a result, there's a lot that is not known about their true distribution. They are both considered data deficient, according to IUCN, as we heard before. And traditional methods of finding them is through local surveys, like just seeing them, observations from the public, or I guess sometimes fishing them up. And on a local scale, seahorses have been shown to fluctuate quite a lot, both in amount, but also in their patchiness in an area. So these traditional methods might not be enough, especially on an international context. As a result, it's good to have more tools in toolbox. And one such tool of surveying is eDNA, which is the analysis of environmental DNA. And basically, every animal which lives in the ocean sheds DNA into the water column as it lives there. And in theory, then, you can take a water sample and say, this animal exists here, if you analyze the DNA there. And the specific type of eDNA analysis I am doing is called species-specific detection, which is when you do these customized primer pairs, which can find DNA from a specific species. And this, along with the method I use, which is digital PCR, can be the most specific way to find aquatic animals. But along with this specific and sensitive method, eDNA is also good because it's quite easy to sample if you only need a water sample, especially in areas that are hard to survey with traditional methods that are deeper, for example. It can detect cryptic animals. For example, these 2 species are easy to mix up, which DNA isn't, and as well as that they are quite hidden in the environment. And finally, this is great because it has such a low impact on the animal itself. There's no disturbance to the animal usually when you just take a water sample in the area. And so the idea was to create these eDNA assays, and that's a bit of a complicated subject. So I tried to break it down into 3 phases, basically. So first, you have the assay design in silico, which is a bioinformatic method, where you create these primer pairs. Then you want to make sure that they actually work in a lab environment by testing them against tissue, for example. And then finally, you can actually test them in the environment to see if these assays actually work to find DNA from these species. And when you create primer pairs, you need to first know which part of the DNA they're going to look after. So basically, they attach to a part of the DNA of the seahorse, and you want to pick a good part, which there's a lot of, and that is specific to the species. And the classic one is CO1, which is the one we looked at. And it was also very good because there was a lot of reference material. There were a lot of these genes already sequenced and available on GenBank. And to make sure that these sequences on GenBank also represented the seahorses we had in Sweden, we actually sequenced one gene from a seahorse that had been found in Sweden. And there's an accession code there if someone's interested. But first, maybe I'll mention the phylogenetic tree. So we wanted to make sure that this gene was a good fit for the species. So we needed to prove that it was specific for the target species we were looking for, but that the gene was also separated between species, if you understand what I mean. We wanted to make sure that we could only find this species using this part of the DNA. And this is a very simplified version of that graph, but it looked really good. It was very well separated. And in the end, when we picked this gene and knew that it would be a good fit, we could generate these assays, which is a method where you use these primer pairs which attach to the DNA. And they were called HipHip after Hippocampus, and HipGut after Hippocampus guttulatus. And with these bioinformatic methods, you can also test them against the database. So basically, we could already, by this point, say that they didn't match anything other than the seahorses we were looking for in the whole GenBank database. However, when you're looking for such a rare animal that is so novel, especially in Sweden, you need to be absolutely sure that it's specific, that it won't amplify DNA from another animal, and that it won't, and that it's sensitive enough method to actually be able to find anything in the water. So therefore, we tested this specificity on tissue samples from different fishes. First, we of course had different individuals of the target seahorses, but then we also tested the method against all the native pipefishes in Sweden, which are 7 species, among with a mock community, which was 37 or so more fish species. Then we also checked the sensitivity, and the sensitivity is basically how much DNA do you need to get a positive detection. And you know, the ocean is really big, the seahorses is very small, so you would expect quite a little DNA in the water. And I'm not sure if these numbers are going to mean anything. Basically, less than 1 copy per microliter would give a really good signal, which were great results. Then we also checked something which was called non-target competition. It's another way that DNA can interfere with each other, and it looked really good for HipHip, and it was a bit inconclusive for HipGut, but generally quite good. And then we also finally checked inhibition. And inhibition is quite common in the environment. It's when different substances actually interfere with amplification. So you have a big chance of not getting anything. And it's not because the DNA isn't present in the sample, but because there were other stuff in the sample that interfered. And it was a pretty simple experiment. We just mixed seawater with aquarium water, and filtered seawater. And we didn't see any inhibition, so that was nice. And finally then, after we had done all of these tests and making sure everything works, we could use it in the field. But we have a big problem. Seahorses are super rare in Sweden. There was no guarantee that they would show up again during the field season. So as a result, we decided to do positive sites, basically take samples where the seahorses actually exist. And these samples were taken in the Ria Formosa Natural Park in Portugal, and in a Spanish site by San Sebastian. And what's even better is that these samples were not just taken where seahorses exist, they were taken right next to these seahorses that were identified to make extra sure that that this method will work in the perfect conditions. And it did. For the Portuguese sites, all samples were positive for Hippocampus guttulatus, which was the species that was identified. One site also was positive for Hippocampus hippocampus, even though it's quite rare in the area, but it does exist there. And the Hippocampus hippocampus individual in San Sebastian was also detected. So that was really good, and we could finally actually test this method in Sweden, of all places. So I went to 7 different sites where there had been previous sightings. And if you look at the graph there, you see that all sites except one, it was negative. And that wasn't surprising. And that's because 6 out of those 7 sites couldn't be connected to a live seahorse for over a year. Basically, a live seahorse was seen in 2024, and I sampled in the summer 2025. So there could be over a year's difference. So it's not surprising that we couldn't detect them. However, there was one site that was special. And if you see here on the picture, it was actually so that the Hippocampus guttulatus showed up on a live camera. And this live camera is situated by Kristineberg Research Station. So I could actually go there the day after it was shown, it appeared, and take samples right next to the camera. So we had both photo evidence of the animal, and then we have photo evidence of sampling where it had been. And yes, this site was positive for the long-snouted seahorse, but actually also a little bit for the short-snouted seahorse. And that was very surprising, because it wasn't seen on the camera, right? However, this site is very close to where one of the 2 Hippocampus hippocampus had actually been seen. It was within 2 kilometers or so.
2 minutes left.
Okay, that's fine. So here are the conclusions. Oh, it's a bit out of order. Okay, so what was created were these 2 eDNA assays, HipHip and HipGut. They can already be used, basically. They work in the field. Especially good probably in areas which are difficult to sample using traditional surveying methods. However, if you want to focus on conservation, you might want to know more about quantifying the amount of DNA. And this method so far has only been used to say yes or no, are they here or not? And for that sake, you need to go a bit deeper. You need to filter bigger volumes. So I only filtered 1 to 2 liters. But to get that amount of DNA you need for quantification, you should try to filter more. You can combine samples, and you can also use this kind of new method called passive sampling, which I think would be really good for these species, because they're only active some parts of the day, right? They're quite sedentary, so you can actually capture the DNA as it comes by. And that was it. So thank you so much for listening. And yes, I also want to add one last thing. Thank you so much for the opportunity of the organizers. This is actually my first conference and my first presentation at the conference. So thank you so much for that.